Thank you all for being here. It's wonderful to be here. This is a wonderful institution. It's my first time visiting. I had a chance to look around, see the roof, see the aquarium and other exhibits, and you've got a gem here, so you should congratulate yourselves. And it's also a real honor to be here helping the Leakey Foundation celebrate their anniversary. It's wonderful to me personally because the Leakey Foundation has always been synonymous with inspiration. And I'll tell you why with a little introductory kind of personal note, which is that, I grew up in Cleveland in the 1970s, working class kid on the south side of Cleveland. There wasn't a lot of inspirational things around me. But the Cleveland Museum of Natural History was a place that I could go. And in the 1970s, of course, Don Johanson was a curator at the Cleveland Museum, and it was in the 1970s when he discovered the remains of this critter, Australopithecus afarensis, better known as Lucy. And there came to be a cast of Lucy in the Cleveland Museum of Natural History. And I remember turning a corner when I was a boy and running face to face, running into this wonderful, wonderful thing and just being captivated by it. And it kind of drove my education and my career. So I went to Yale and I had every intention of becoming a paleoanthropologist, and I spent several years studying paleoanthropology. But one of the things that became clear to me, and I think it's been clear to a number of people, even going back to Louis Leakey, is that if you want to understand that, you really need to understand the behavior that produced those bones. So you can't understand the markings, the ridges, the size, the shape of the bones that you dig up unless you can understand something about the way that relates to muscles and the way that the muscles relate to behavior. Okay? And then I came to the realization as a graduate student that if I want to understand behavior, actually I really need to understand the brain. I need to understand the way that the nervous system actually produces behavior. Okay? So that has been kind of the story of my career and why I am where I am today, kind of a jack of all trades, master of none, studying this intersection between primatology, evolutionary anthropology, social sciences, and neurobiology. Okay, so what are we going to talk about today? Well, today we're here to talk about this. We want to talk about innovation. How in the world is it? This is something that's really kind of unique. We would like to think of it as uniquely human. We're characterized by our innovative natures. We are so dependent on technology and improvements and the creation, really, of new technologies is happening at an incredible pace. Right? We can't imagine our world without all of these amazing devices. But if you think about it, innovation is something that's tricky. It's something that's unusual, right? Something that's unusual in the animal kingdom because it means stepping outside the box, right? You know something that gives you a little bit of a return, gives you a little bit of food. And most animals are really good at this, right? Through learning, they learn to associate behaviors that are rewarding. Okay, innovation, creativity, exploration demand stepping outside the box. You have to risk uncertainty and look for something new. It's really a fundamental characteristic of the adaptation of our own lineage, right? So moving out from the trees onto the savanna, developing stone tool technologies which really pave the way forward for our advancement as an organism. As a clade, really depends on this capacity for innovation. Clearly, it derives in some measure from our simultaneous tendency to want to explore. Right? Exploration is part of what it is to be human, and in particular, I think, part of what it means to be an American. It demands, as I mentioned before, some kind of tolerance for uncertainty. Right? When you are exploring, when you are innovating, you don't know what's going to happen. It might demand a little bit of self-control, right? So you might have to be able to delay your gratification in order to see what might happen next. And I think one of the things that's often overlooked is that innovation benefits from other people being around, right? I think Google knows this, Apple knows this, right? Facebook knows this. So this collision with other individuals can generate new ideas, right? We bring different things to the table, and we can also learn by observing what happens to others. America in particular, right? This is our nature. We're an immigrant culture, and perhaps it's by no surprise we're the most innovative that's ever inhabited the planet. So what does neuroscience have to say about innovation? Well, this is something that has become something of a hot topic of late, but really wasn't one that was open to exploration more than 10 or 15 years ago, and that is due to the fact that we now, over the past decade or so, have the ability to image, to measure activity, blood flow activity in the brains of awake, living human beings, okay, using typically functional magnetic resonance imaging. So people, if you've ever had an MRI to get your knee looked at, you know, you've got arthritis or something like that, it's the same procedure, except you go lie in the tube and your head's being scanned. And you can use basically you get a really smart physicist to come in and to help you program the MRI machine so that rather than just measuring the structure of the brain, you can measure something about its activity. Okay, and I'm sure if any of you pick up the New York Times or you look at anything online, you get to see these pretty images like this. Beautiful, the human brain in action. And what we've learned recently, and so this just to orient you, this is the left side of the human brain, the eyeballs would be right here. This is the midline of the brain, now the eyeballs would be here. This is the top of the brain. This is the right side facing that way, okay? Is that all clear? And one of the things that's really been very interesting, there have been a number of these studies that have been done recently, just looking at the human brain while you're doing nothing. Okay, sounds kind of weird and interesting, but when you're doing nothing, there's this particular set of brain areas that are highlighted in green, which are incredibly active. In fact, they chew up more energy, they chew up more oxygen than any part of your brain. And the hypothesis is that these parts of the brain seem to be involved in mind wandering, thinking about the future perhaps, who knows, thinking about basically setting your strategy for how you're going to deal with the world. And these areas seem to be in direct kind of competition with other areas here, which I'm just highlighting a couple of them, which are highlighted in yellow and red, which are most active when you're focused on something that you know how to do, right? When you're really focusing attention on, you know, whatever it is, you're cooking dinner or you're working at your computer, but you're not aimlessly letting your mind float around. Okay, these areas are strikingly active. And then there's another set of areas here which is involved in planning. Now I just highlight this other area in the frontal lobe, which is involved in planning ahead. Now, one of the things that neuroeconomics has begun to teach us over the past 10 years, this is a new field that really is the marriage of the mathematical formalisms and behavioral approaches from economics with contemporary neuroscience aimed at understanding the biological basis of decision making, is that all of these areas kind of interact in a way whenever we do make decisions in a complex way. And in particular, these areas, all of them, both kind of the mind-wandering areas and the attention and focusing and learning areas are engaged when you're dealing with other individuals. Okay, so that's what I want to talk about today, how all these areas seem to, at least I'm going to give you a glimpse of how they might be interacting and how they might be shaping the ways in which we can think creatively, explore our options, learn from other individuals, and sort of strategically plan in competitive contexts. Okay, to come up with new ways to behave. Now, all of these great neuroimaging studies rely on this functional magnetic resonance imaging technique, which, although it produces beautiful images, is rather crude. So it is a sluggish signal that evolves over the course of 6 to 8 seconds because it's relying on blood flow measurements. And in each one of these areas that is highlighted like this is encompassing hundreds of thousands, if not millions of neurons. Here's a nice drawing from one of the early neuroanatomists, Santiago Ramon y Cajal from the 19th century. So showing basically that within these hundreds of thousands or millions of neurons, they may have different shapes, different morphologies. They may receive projections from different areas. They may project to different areas. They may express different receptors. So we have every expectation that different kinds of neurons within these areas might be doing different things. They might be subserving different purposes. And that although that brain imaging signal from humans is wonderful in pointing out something about what might be going on in the brain, we've got to dig a little bit deeper. And that's kind of where my work comes in, because what we are doing is advancing techniques for trying to understand how the activity of these neurons, how the chemical messages they're sending, how the receptors they express might actually contribute to the computations that produce behavior. And what I'm pointing out here is this is the brain of a rhesus macaque here on the left and the brain of a human. They're not to scale, of course. The human brain is much, much bigger than the macaque brain. But the main point I want to make here is that all anatomical and histological studies that have been done basically show that there aren't any really new areas in the human brain. that basically monkey brain has basically the same brain areas that a human brain has, and they're connected up in roughly the same ways. So what we are walking around the world with is a 30-million-year-old device. Okay, it's built, to forage for food, to gather information, and to deal with others. We may do that in fundamentally new and interesting and complex ways, but we are tapping into primordial circuits. And the way that we approach this in my lab is thus. We begin by thinking about the problem that the organism has to solve in the real world, whether that's a human or a rhesus macaque, dealing with others, exploring for new opportunities, etc. We try to translate that into tasks that we can implement in the laboratory, that resemble, or at least bring into the laboratory, the kinds of information processing problems that the organism faces in the real world. And then we can use various techniques in the laboratory, whether they are making fine-scale recordings from individual neurons in the brain, doing complementary brain imaging studies in people to see how they match up. We can do pharmacological studies, such as, for example, manipulating oxytocin levels in monkeys or in people and seeing what effect that has on behavior and has on brain circuits. And then one of the things that's been really exciting in the past few years is that we've taken this full circle and we've now gone back into the field and we have begun to explore the ways in which naturally occurring variation in social behavior and decision making, in cognitive flexibility, actually has a biological basis. So here is a map of the social network in one group of rhesus macaques on the island of Cayo Santiago off the coast of Puerto Rico. These are monkeys. This population of monkeys has been there for 75 years. They've been under continuous observation that entire time. And what we do is we basically have people, it's tried and true anthropology or ethology. Basically, you follow around an individual monkey for 10 minutes at a time, or you record everything that they do. But from that data, you can construct something like this, which is a map of all the social relationships. And I'll get back to this later, but we can map. Basically what this shows is that some individuals are really tightly integrated into the social network, and some are very peripheral, some are completely peripheral. And we can map, for example, gene expression. We can map the genes that the animals actually come into the world with, their genotypes, onto this, and in those ways begin to figure out how an individual's biology and the circumstances into which they're born shape their ultimate expression of behavior. We can then take that information back into the laboratory and figure out how it works. That's the basic research program. So what I want to talk to you about today is three things: creative exploration, learning from others, and innovative competition. I want to provide experimental evidence that, like humans, rhesus monkeys engage in all of these kinds of behaviors, and that we have begun to work out some of the underlying neurobiology, which I think points the way forward on understanding our own neurobiology, and also may help inform us on potential therapies for those individuals who have problems in any of these domains. Now, as I mentioned before, there's this interesting network of brain areas. It's often called the default mode network, which when people are kind of doing nothing is highly active. It includes sort of parts of the posterior cingulate cortex, parts of the ventromedial prefrontal cortex, and the lateral parietal cortex. As I mentioned before, activity within this area, kind of hemodynamic blood flow related activity, seems to vary with one's tendencies to let their mind wander. And in some cases it might predict how creative you are, how many different words you can come up with, how many different functions for a novel object you can come up with if kind of given that test. So what's going on in these areas? Do neurons themselves actually spike? Do they fire? Do they signal information in a way that reflects this kind of activity? And we have examined that in monkeys, and here is kind of a summary of that. So what I'm plotting here is how much these neurons are firing relative to baseline in different conditions in monkeys. So the baseline level is here. And what's really interesting is that when the monkeys perform a task, like a memory task or an attention task that they know really well, they've done it thousands of times, literally. And monkeys basically do this to get squirts of juice. They love fruit juice. We figure out what kind of juice they really love, and each monkey gets his own individual fruit juice. So whenever the monkeys do a task that they really know how to do, it's very repetitive, we see these neurons shut down, just as the hemodynamic signal, just as this functional imaging signal in humans would lead us to believe. But one thing that's really interesting about this is that the levels of activity vary with performance. So when monkeys are really good, they do the task really quickly, you see a a greater suppression. And when they're worse at it, when they perform more slowly, we see less of that suppression. What's even more fascinating is that whenever the monkeys make an error, you can tell they're going to make an error well before they do it because you see activity go above baseline, as if they were kind of letting their minds wander, and that's when they made an error. Now, what's really kind of cool here is that occasionally we give the monkeys a cue that says, take a rest, don't do anything, right? And when we do that, we see that, in fact, activity goes up even higher, right? Now, we don't know what the monkeys are doing then, right? But we can only imagine that they're not thinking about the task that they have to do. They're not thinking about the problem that's in front of them. They're probably thinking about, well, what am I going to do when I get back home after I get off of work? I wonder what everybody else is doing back in the colony. This is purely speculation, but I think it's an interesting speculation. Now, one of the things that we've gone on to do, of course, we're neuroeconomists, we want to understand decision-making, is we've looked at these neurons, the activity of these neurons, in other kinds of conditions. And there's something really interesting there, which is that not only do these neurons, their activity vary with kind of cognitive state, but they also vary with what the monkey is experiencing. So if you give the monkey a reward out of the blue, we see a strong response from these neurons. And in fact, firing rates vary quite lawfully with the amount of juice that the monkey might get in a surprising context. So the more juice it gets, the more neuronal activity goes up. And these neurons are also sensitive to uncertainty, right? And remember that in the beginning, we talked about the fact that exploration, innovation demands some kind of sensitivity to and an ability to overcome uncertainty. And we see that in a task where the monkey is choosing between a sure bet and a gamble on double or nothing in terms of the size of a juice reward, that whenever they're going to take the gamble, we see increased activity in this area. When the monkeys are instead taking a gamble between basically a small reward now or an uncertain delayed reward that could be really big, we also see an increase in activity, even though they generally hate to wait for a reward, and they'd much prefer to have a smaller reward sooner. And we also see that when they are taking a gamble on the possibility that a picture of another monkey could come up, but they don't know who that monkey is going to be. Okay, we also see increases in activity. So in all these conditions, right, that are uncertain, when there are potential rewards to be harvested, okay, but you don't know whether you're going to get them, we see an increase in the activity of these neurons that we know their increases are also associated, or we think are associated with exploration and mind wandering. And in a task in which we actually presented the monkeys with an opportunity to explore their options, they're basically tracking the value of 4 different slot machines, okay? If they're choosing one of the slot machines, they know exactly the payoff, but the payoff is going to wander on each trial. If they're not, the payoff for the other wanders as well, but they don't know what that payoff is unless they choose it. So basically what they do is they tend to sample amongst them, they find one that's good, and they stick with it for a while. But as the payoff starts to decline, they might start to think, I bet bet another one of these slot machines is better now. Right? In fact, we do see that mathematically they switch, they explore, they enter in a strategic mode of exploration when they ought to, kind of based on the mathematics, okay? And what we find is that if we're recording from these neurons, this is across a population of neurons in this default mode area, that neuronal activity is slightly but significantly higher throughout the course of trials when monkeys are about to engage in strategic exploration. Okay, this is not a time when they are trying out their opportunities, moving away from a sure bet, okay, that they know what the payoff is, and hoping for the best. Okay, this is an opportunity to learn. This is an opportunity to try new things. Now, I'm going to be able to tell you here that we can go beyond mere correlation, and I can demonstrate to you the fact that we can show a causal role for this area, for these neurons, in motivating exploration behavior, because we can now, instead of listening to these neurons, we can artificially activate these neurons using very small amounts of electrical stimulation. And when we do so, basically what we do is we cause monkeys to explore an option when they otherwise wouldn't have. So in a simple gambling task, the monkeys tend to favor the risky option. They like to take the gamble. If they get the jackpot, they're going to go right back to it, right? Just like a human gambler. If we stimulate these neurons after they get the jackpot, the monkeys are much more likely to switch to the safe bet, okay? So this provides some direct causal functional evidence that these neurons are involved in creative exploration. So now I want to turn to the next component of innovation, and that is learning from others, being in an environment where we can learn things from the experiences of other individuals. Now, we all know from our own lives that we learn from others, and this capacity to identify with the experiences of other individuals, of course, we call empathy, or at least that's sort of the emotional component of it. And neuroimaging studies in people have suggested that parts of, this is sort of the middle view of the human brain, so if you kind of sliced my head down the front here and pulled off the left side, you would be looking at this. And what's shown here is are the responses of this area when people are seeing another person in pain. Okay, you can also do this if you show your subjects people who are experiencing rewards. And basically what you find is that this area becomes active when people are identifying with the experiences of another individual. And across participants, so each of these dots is a different person in the test, the amount of activity you get here in this area called the anterior cingulate cortex scales with how much people identified with the experience of the other person, suggesting that if you are a person who really empathizes with somebody else, this part of your brain is really actively engaged. And people who don't empathize with other individuals, who maybe don't see them as people or don't think about their experiences, you don't see that kind of activity. Well, we wondered whether, first of all, whether we might see similar kinds of sensitivity to the experiences of other individuals in monkeys, and what parts of the brain might actually be involved in supporting this behavior. So this is a bit of a leap for me. This is actually a set of studies that I've been wanting to do for about 15 years, but we never felt like we were quite ready, that we needed, the science hadn't advanced enough yet, and that we're not ready to really study the way that individuals interact and experience what's going on during a dynamic social interaction. But after getting tenure, I finally felt like I had the confidence and the security. Don't get rid of tenure. You need a little bit of a secure base to take on crazy experiments like this. So here's the basic setup. Rather than having one monkey looking at a computer screen, we have two monkeys who are sitting there next to each other in the experiment room. They're sitting at about 45-degree angles to each other. These are monkeys who know each other. They each have their own computer screen that they're looking at. They each have a juice tube in front of their mouth that they can get squirts of juice. And we also are recording the position of their eyes with a very high-speed, high-accuracy infrared video camera. Okay, so we know to the sort of degree and with millisecond precision where they're looking. And we also are recording from the brains, at least of this donor, from neural activity in their brain, or we can also manipulate the chemistry of their brains in the same kind of way. So basically you can think about, if any of you have teenage kids, how many of you have teenage kids? Anybody? So I don't know if you've ever noticed the fact that they can be sitting next to each other and they're texting each other. So they're not actually talking to each other. They're writing to each other online, either text or email, and occasionally look over at each other and laugh, but they're not actually talking, right? So this is kind of what our monkeys are doing. And here's the basic game. Over the course of a few days, the two monkeys are basically just seeing colored shapes on the screen. Okay, there are a couple of different conditions here, but the details don't really matter. And what's important is the way that the colored shapes predict what happens. So one colored shape, like a red box, might come on, then there'd be a little delay, and then the donor, what we're calling the donor monkey, might get a squirt of juice. Okay? A green box might come on, and the recipient monkey would get a squirt of juice. I don't know, blue box might come on, and both monkeys get a squirt of juice. Hooray, that's the best of all. And maybe a white box comes on, and nobody gets a squirt of juice. But the monkeys still hear the sound of juice coming out of a tube in the other room. Okay? And over the course, so nobody got reward, but we want them to know that there was an outcome, right? So they basically do that. And then, of course, we assume that the donor monkeys, of course, care about when they're getting juice, right? And we know that. Monkeys work hard to get juice. But the interesting question here is whether what happens to the other monkey alters their experience of juice, right? So does the other monkey getting a squirt of juice make your own feeling of juice feel worse, right? Do you have some competition there when you're both getting a squirt of juice? And even more interestingly, do you care when the other monkey gets a squirt of juice, and is that better than nobody getting a squirt of juice? Okay, so that's actually the critical conditions. So if we examine, say, a choice between reward to the other guy, reward to nobody, the donor monkeys never had a squirt of juice on any of those trials, right? And if all he cares about is his own reward, he should just not care, right? He should not have a preference amongst those two choices. He might not even want to work. He might just say, I don't care. I'm not going to do this trial, okay? Similarly for the other choice that is matched for reward to him, reward for me or reward for both of us? In both cases, he gets a reward, so you might predict if what happens to the other guy doesn't matter, then he will have no preference under those conditions. Well, I'll show you that, in fact, they do have preferences under those conditions, and they're very systematic, and they're pretty profound. So what's plotted here basically is the results of the behavioral choice data. This is in the context where it's either a reward to the other monkey or reward to nobody, and when the bar is going up, that means more likely to take the pro-social choice, and this is a logarithmic scale, so it's actually a pretty strong bias. So we can see that these are a number of different pairings of 2 donor monkeys and a bunch of different recipient monkeys, and across all these monkeys, basically, there's a preference to reward the other guy versus nobody. So they care what happens to the other guy, right? It's meaningful to them, despite the fact that these are rhesus macaques, right? These animals are known for being despotic, competitive. They can be very aggressive. But of course, if you think about their natural history, there's an importance to maintaining strong social bonds with other individuals, having friends and allies who can help you out in a fight, who can help you to defend your territory, right? And we think that's what this is unmasking. Now, critically, you might be thinking there. If you're thinking critically out there, you might say, well, maybe they care about the other guy, but maybe they just like seeing juice come out of a tube. These monkeys love juice. Maybe it's just enough to see juice coming out over there, right? So if we instead remove the other monkey and replace him with a bottle that collects the juice, now our donor monkeys don't care. They have no preference. So it is critically dependent on the presence of another social being, right? That's what this depends on. Now, what's kind of confounding here and kind of makes your head spin is that when it's a context between juice for me or juice for both of us, now they're selfish. They say, I want it all, right? So we think that is because they interpret this as a competitive context, as if they were drinking from the same puddle, right? And they don't like that, right? They are competitive monkeys. At the same time that they are cooperative and they form these strong social bonds, they also compete with each other, and that's what's revealed right here. Again, if you remove the other monkey and it's replaced with a bottle, now they're indifferent. Okay, so it's all about the other monkey. Now, of course, we hope that this depends on, if this is really a social interaction, then it ought to depend on the relationship between the 2 monkeys, and it does. So what we found is that monkeys, our donors are more benevolent to monkeys with whom they're more familiar, and they're less competitive with them, so that's blue versus green bar. And monkeys who are dominant are more benevolent and less competitive when the recipient is subordinate. Okay, our subordinates are very selfish. Okay, but that's only in a situation where they're confronted with a dominant individual. That could be because that situation is very stressful. It could be because they finally have the opportunity to screw the other guy, right? I mean, who knows? We don't know. It's speculation. Okay, so what's going on in the brain? Well, we have studied the activity of neurons. This is kind of what's called a coronal section through the brain. So if you were just kind of open me up here and look on face. We record from neurons in the orbital frontal cortex, which I mentioned before is kind of involved in reward processing, involved in this kind of exploration behavior. Parts of the anterior cingulate sulcus, which is involved in learning and attention, and also this other really interesting area, the anterior cingulate gyrus, which, as I mentioned before, we know in humans seems to be active when you are empathizing with another individual. And the results were pretty telling. So this orbital frontal area, which is involved in processing rewards, we know it. These neurons respond when you see another individual, when it's an individual you like. Nonetheless, these neurons only respond here, both of these red colors, when the actor monkey chooses to reward himself, when he gets a physical reward. So this is about the direct personal experience of reward. Okay? The neurons from this more dorsal anterior cingulate sulcus area are the exact opposite. They only respond whenever the actor monkey decided to give away a reward. So these are all about foregone rewards, and we have in previous studies linked activity in this area to regret, to thinking about what might have been, the kind of woulda, coulda, shoulda, if only I had chosen something else, this is what I would have had. Okay? Now the most interesting neurons are here. These are neurons in this area that has been identified with empathy in people, and we find that neurons there respond both when the monkey gives himself a reward and when he gives a reward to another monkey. Okay? As if these were equivalent experiences, right? So we've turned these neurons mirror neurons for reward. It's as if they are incorporating the experience of the other individual into their own response. And we've also found that the fidelity with which these neurons signal information predicts how pro-social the monkey will be. So when these neurons are really doing their job well, the monkey is much more pro-social. And when these neurons are kind of not doing a good job, they're not firing very accurately, then the monkey is not very pro-social. Now that's not a great test. I'm just going to move on from that. In a more recent study, we have been examining how monkeys actually learn from observing the experiences of another individual. And in this study, basically what we do is we do a little chemical cookery. So I have undergraduates in the lab who every day make little gelatin capsules, and they can be any arbitrary color and any flavor of juice inside. So it can be yellow associated with blueberry juice, or red, and it could be tonic water. You know, I like tonic water. Monkeys don't like it. It's very bitter. And then basically, we have one monkey who's an observer and another monkey who's a demonstrator. And so the observer monkey watches the demonstrator try these new foods every day. And when the demonstrator picks up one of these little jelly beans and eats it, he's either going to go, yum, or he's going to spit it out and make a face. And our observer monkeys learn very readily from watching the demonstrators. So even if they see that demonstrator reach out for something that they saw him eat before and like, the observer starts licking his lips as if he is about to taste it. Okay? Now, one thing we found is that we can turn these neurons in these reward mirror neurons off with a short-acting drug that increases inhibition within that part of the brain. And when we do so, I just want you to see what's going on here. This is social learning and non-social learning. Non-social learning is learning from your own experience. Social learning is learning by watching the other monkey try the foods. When we shut this area off, which occurs right here, and we switch the color flavor pairings, from direct experience, the monkeys pick it up quite readily. So it goes from being bad to good, and now they start looking at it more, and they lick their lips more, etc., and they stop selecting the bad one, but they can't do it when they're watching the other monkey. Okay, so these reward mirror neurons, which incorporate the experience of another monkey, are critical for actually learning from what that animal experiences. And we think that's probably true for people as well. So now I'm going to turn in this next section to the possibility that innovation also springs from competition. Okay, I think we hear that all the time. This is a capitalist country, after all, right? This is something that we ought to expect. And of course, living in a society with lots of other individuals, of course, there's lots of opportunities for the things we just talked about: empathy, trust, compassion, charity. But there's also room for this, which is, anybody remember this guy? Okay, Bernie Madoff. Strategic manipulation, deception, right? Using other people's trust to your own advantage. Okay, that's an innovative behavior, if ever there was one. It requires a lot of planning ahead. You kind of got to be unpredictable and predictable at the same time. There's been very few neuroscientific studies of deception, but what studies have been done are pretty interesting. We do see activity within the anterior cingulate cortex here. Okay, this is the midline again, the front of the brain, right? The part of the brain that we think is involved not only in learning, but perhaps empathy and modeling the experiences of other individuals. But we also see activity out here in the lateral prefrontal cortex. This is a part of the brain that's involved in strategic thinking, long-term planning ahead, right? These kinds of behaviors. And so building on the success of our more recent dynamic interactive studies in monkeys, we thought, well, maybe we could study deceptive behaviors in monkeys as well and see if we can work out the underlying neurobiology. And so that's what we've done. We've developed, this is the work of a very talented postdoc in the lab, Jean-Francois Garipé, and he's developed a beautiful study in which basically we have the same setup where two monkeys are sitting next to each other, but rather than having them decide whether or not they're going to give a reward to the other monkey, they're playing a computer soccer game. Okay? And so we call it penalty kick. Here's the basic idea. One monkey's controlling the ball, the other monkey's controlling the goalie. If the monkey who's controlling the ball gets it past the goalie, he gets a squirt of juice. If the goalie blocks it, he gets a squirt of juice. And we titrate the size of the goalie, so basically the two monkeys win about the same percentage of the time. And they switch roles. So the monkey who is the kicker will be the goalie on the next trial, and that's a great shot right there. Right? The thing that, so what's and then they vice versa, they switch their roles. And one of the things that we noticed right away is initially the kickers start out playing very simply. They make basically straight line kicks. Right? Think about kind of your typical penalty kick if you're watching a soccer match, this sort of straight line kick. But very rapidly they learn to make these very circuitous, seemingly very deceptive movements. Okay? It's really quite striking. And we think this is a socially motivated behavior. We track the eyes of these monkeys. Here are recordings of eye traces, basically where the monkeys are looking while they are playing the game in one day, where the kicker's looking. You can see this is the computer monitor, and he's looking initially at the ball. And of course, the trajectory of the ball spreads out. He's looking at the goalie. But he's also looking at his opponent's joystick. He's looking at his opponent's face and his eyes. He's trying to predict what the other monkey's going to do, right? So he's trying to gain an advantage there, and this is social. Now, when we're training them, we test them both against a live goalie, and we also test them against replays of the goalie playing yesterday, right, to see if they can tell the difference. And they can. So here's one-day sessions where the monkey's playing against a recorded goalie, and I hope you can see the fact that he rapidly recognizes that. I'll show it to you again. And he starts becoming very predictable. He's like, oh, the other guy's not responding to me, right? He's not really trying to block me. I can just go to the same place every time. Now compare that with playing against a live dynamic goalie. Incredibly unpredictable, right? In fact, they don't really start making moves until the very end. And that's just kind of summarized here. Against a computer opponent, against a replay of another monkey, and against a live monkey opponent. So you're much more unpredictable and seemingly deceptive. And in fact, we classify many of the movements, many of the plays that our monkey kickers make as tactically deceptive when the monkey kind of basically goes in one direction but ends up at a different point in his goal. And using kind of some mathematical information analysis, we can show that our monkey kickers are incredibly unpredictable. They're hiding information about their intentions until the very last second. So they're kind of going, and then they end up somewhere where they weren't going to be. It turns out that they actually look ahead at the very beginning of the trial. They look at where their goal is going to be. Now, we've recorded from neurons both in this dorsolateral prefrontal cortex area that we know is involved in strategic planning in people. And when monkeys are playing the game, we see something like this. This is activity of one neuron. In red is when the monkey's going to fake out the other monkey by going, eventually ending up at the upper part of the screen. And we can see a big burst of activity here, and it happens well in advance of the time that he actually makes the fake movement. Okay. When he makes a downward fake or when he makes a straight play to the upper part of the screen, we don't see that activity. Now, importantly, we use a control condition in which we basically ask our kicker to move the ball through a set of hoops to get to the same goal. So he's performing the same action, constructing the same plan, but now he's not doing it to deceive the other guy. He's not doing it on his own, and we see no real selective responses on the part of these neurons. We've also recorded from neurons in the more medial part of the prefrontal cortex, okay, also active in people. And I just want to draw your attention here. This is the time of the last, of the fake. And we don't see any selective activity until after the fakes have occurred, and what these neurons seem to be signaling is just basically the very last movement. Okay, so it's a very different kind of area, and if we classify all the neurons, the ones in the lateral prefrontal area, this planning area, seem this 31 of these neurons out of our sample, almost 50% of our neurons, seem to be selective for strategic deception. Okay, and we just don't see those neurons in this other part of the brain. Now, what's really important is, again, we can make a functional analysis here. We can look at the causal contribution of these neurons to playing deceptively, to engaging in this kind of unpredictable, exploratory, innovative behavior. And we see that in this medial prefrontal area, if we increase inhibition with a drug, there's no difference in the behavior of the monkey. If we increase inhibition in the dorsolateral prefrontal cortex, what happens is the monkey becomes much more predictable. This is a mutual information measure which just measures how predictable the monkey is. So now he can't fake the other monkey out. The only thing he can do is more or less make straight plays. He can't develop these long-range strategic plans. Okay, so I conveyed to you some of the work we've done demonstrating experimentally that monkeys engage in creative exploration, learning from others in innovative competition, and we've begun to tease apart some of the underlying neuronal mechanisms and to show that they functionally contribute to the behavior themselves, either by manipulating the activity of these neurons, either activating them or shutting them down. Now, one of the things that we have become increasingly interested in studying decision making, studying social behavior, is the fact that not everybody behaves the same way, right? So some people are more creative than others. Some people are greater risk takers. Some people empathize more with other individuals, learn more from other individuals. Some individuals are more deceptive. So what is the root of these individual differences? So let's take these two guys here, familiar to everybody in the Bay Area. This guy, both are entrepreneurs. This guy typically associated with not very innovative products, let's say, kind of the not cool guy. This is the guy we think of as being very cool, producing these wonderful, innovative products like my laptop here and the iPhone in my pocket, etc. But if we think about their social behaviors, we know that this guy wasn't too nice, right, to the people around him. Not a great philanthropist. This guy, one of the world's great philanthropists, right? So there's something fundamentally different about them. What is it about them? What was it about them? It's about their experiences growing up. Was it about something about the biology that they came into the world with? And that's an intriguing, important, fascinating, difficult question to answer for people because our own behavior is so complex, right? It's very difficult to actually study this in an empirical way, although we are doing it. Other people are doing it as well. We have been trying to answer this question in a project with rhesus macaques where we think that it may be a little bit more tractable. So where we can gather exquisitely detailed data on the behavior of these animals, their phenotype, right, in a situation where the animals don't care that we're observing them. So when we try to analyze the phenotype of an individual person, we bring them into the laboratory, they answer a bunch of questions on a questionnaire, maybe they play a computer game, but that's not the same thing as seeing what they do in real life. But I don't think people want us following them around, recording everything they do. The monkeys don't really care. So here's the way that we've been doing this. We have been studying a population of over 1,000 rhesus macaques on this island of Cayo Santiago off the northeast coast of Puerto Rico. They were transported there 75 years ago. We basically know everything about this population. They've been under continuous study. We have access to incredibly rich and detailed demographic and life history records, records of their dominant status, when individuals migrated from the natal group to another group, who had more babies, who was dominant at what time. And then we are, of course, gathering all this wonderful behavioral data and some experimental data as well. And I just want to give you a teaser about the kinds of things that we can begin to explore. So from our detailed behavioral observation data over 2 years of a single group, Group F on Cayo Santiago, we can use social network analysis to look at the ways in which individuals are bound up into a society. Okay, this is a mathematical graph theory-based analysis, and this is for 2 different years. And what I'm showing you is the network of grooming relationships. So think of grooming as the major way in which non-human primates build positive social relationships with each other. Think of it as an investment in another individual. If I groom you now for 20 minutes, maybe you'll aid me in a fight later. We also looked at the ways in which they fight. Rhesus macaques fight a lot. You can see there's lots of connections here. And also just kind of where they sat. These are just kind of 3 basic behaviors, but we can examine more. And there's a lot of richness to this. So the squares are females and the circles are males, okay? And the connections reflect kind of the frequency and strength of the interactions between those individuals. And one thing that's apparent here is that there's some individuals who are in the center. They have a lot of friends. They have a lot of connections. They have a lot of allies. And some individuals are on the periphery. Some of them have only one friend, okay? And there are some individuals who have absolutely no friends at all, no connections with anyone. They get in fights, but they don't have any friendships. They don't have any positive social relationships. So what is the cause here? Well, surely it's complicated, but we have begun to map these social relationships onto an individual's genetic heritage, okay? And so we have found that where individuals are within, say, the grooming network is heritable. So if you are a female who's kind of in the center, if you're very popular, you have a lot of friends, your offspring are likely to as well. Even your male offspring who are going to go off and join a different group, okay? They are likely to also have that same mix of personality and skill. We don't know what the components are yet that will allow them to thrive and be very social. If you are on the periphery, if you don't make connections very well, if you have offspring, then they'll kind of share some of those traits. It turns out that individuals who are in the middle here have a lot more babies, they have higher reproductive success, they're biologically more successful than these individuals out here. Now, what's the kicker here is that we've begun to look at the genetics of these behaviors, just in a very, very simple way right now. So in this case, we're just looking at variation in genes that are involved in serotonin function. We know that serotonin is involved in regulating mood, it's involved in depression and anxiety in people, proneness to addiction and alcoholism, etc. Within this group of monkeys, those individuals who kind of have 2 copies of, or a copy of the bad allele at each of 2 genes that are involved in regulating serotonin function tend to be on the periphery here. So those are the individuals in red, and they have significantly fewer connections, and the connection strength with other individuals is significantly lower than those individuals who have basically good copies of those genes, who we can think of in kind of a crude sense as having a very well-functioning serotonin system. And this is really fascinating, right? It suggests that there's a real adaptive advantage that is genetically conferred. We're kind of looking at evolution happening, and the question that should immediately pop to mind is, why do we see any of these red guys at all, right? Why are there individuals who are carrying around this kind of bad, so-called bad genes? So we speculate, and others have speculated too, that of course serotonin might be involved in regulating positive social relationships, but it also might be involved in other kinds of behaviors that might be advantageous under other conditions, like being a risk taker, right? Like being socially wary and anxious. So if there's a lot of turmoil, maybe it's better not to be in the mix if there's a major social upheaval, like we just saw actually last year in this group. This group had a major social upheaval and it split into 3 groups. And all 3 groups are doing terribly, right? They should have stuck together, but they couldn't handle it. So these are ongoing studies. These are only 2 genes. We are currently involved in basically sequencing the entire genome of all the monkeys on the island. We want to make this a publicly available resource that can inform lots of other studies. So I want to wrap it up here, and I hope that I have at least planted the seeds, if not convinced you, that there's an importance to understanding the underlying biological circuitry that allows for behavior, in particular complex social behavior, learning from others, exploration that can lead to innovation and perhaps lead to the emergence of new forms of life that, in our own case, right, has had a transformative effect on the world. So with that, I'll thank you all, and I really want to thank all the people in my lab group who have done a lot of this work, in particular Steve Chang, who's a postdoc who's moving on to Yale, who's done all of the kind of empathy-like studies. Ben Hayden and Sarah Heilbronner did all of the creativity and exploration studies, as well as John Pearson. Jean-Francois Garipé has done all the deception work and social learning work, and Lauren Brent, she's a primatologist extraordinaire, and she is heading up our field efforts. So thank you.